Yes, currently we will study about metaphase and transition to metaphase. In our last lecture, we had covered about the prophase. I'm just giving you quick review. Amphase was the most dramatic phase. In case of mitosis, number of chromosome will remain same. So that's why mitosis is also known as equational division. In case of mitosis, there are two stages, that is karyokinesis where nuclear division will occur, and one is cytokinesis where cytoplasm will divide parent cell into two daughter cells. In karyokinesis, there are four further stages, that is prophase, metaphase, anaphase, telophase, and in NCERT it is written there can be no clear-cut lines which can be drawn to explain the end and the beginning of the another phases. Prophase is the first stage of the karyokinesis where chromosome starts their condensation, and during condensation they look like ball of wool. They look like ball of wool, which is known as the spiring stage. Then chromosomes during condensation from chromatins, they form chromatid like structures. They form chromatid like structures where one chromosome has two sister chromatids attached to one centromere. For our convenience, we divided prophase into early prophase, mid prophase, and late prophase. In early prophase, I explained about the condensation of chromosomes in my previous video, and in mid prophase, there was the ester formation. With the ester formation, centrioles moved toward the poles. It was characterized by bolward movement of the centrioles, and then later we studied there are two ester form in case of animals, but in plants there is no ester as there is no centriole. So division of plants is known as anaestral, and in animals division is known as amphiestral, right? Later in the late prophase, nuclear envelope disintegrates, and due to disintegration all the nucleus content gets separated into the cytoplasm, and cytoplasmic organelles get disappeared. These were the NCERT diagrams. And one important note point, prophase is the longest stage of the amphase. In case of karyokinesis, there can be one question in your NEET, which is the longest stage of amphase. The prophase is the longest stage of amphase. So in this lecture, we will study about the transition to metaphase. When prophase ends, before entering to metaphase, there is a phase which is known as prometaphase, or a transitional phase from prophase to metaphase. In this phase, there are spindle fibers which can be clearly seen. This point here, this white point is known as kinetochore. This point, this disc-like structure is known as kinetochore. NCERT does not explain much about prometaphase in the text part, but it is explaining it diagrammatically. This is your NCERT figure. This is the kinetochore, which are the disc-like structures and the attachment sites of the spindle fibers. These are present on the lateral sides of the centromere. This is one chromosome having two sister chromatids, 1 and 2. Here in this stage, before entering to metaphase, from these centrioles, these are perpendicular pair of centrioles. From this centriole, spindle fiber arises, and these are attached to the kinetochore. From this centrosome, this spindle fiber, and from opposite pole, opposite pair of centrioles, this spindle fiber. That means per chromosome, two spindle fibers are attached, and these spindle fibers that are attached to the kinetochore of the chromosome, these fibers are known as discontinuous fibers or the kinetochore fibers, and one more name is tractile fibers. This spindle fiber which is coming out from this centrosome and is attaching to the kinetochore site is known as tractile fiber, discontinuous fiber, or the kinetochore fiber. So in prometaphase, these spindle fibers comes out and gets attached to the specific sites, and also this pair of centrosome is attached to each other. This pair of centrosome is attached to each other with the help of one spindle fiber which is known as continuous fiber that attaches one pole to another pole, pole to pole attachment. So we can say what is happening in the transition to metaphase. Spindle fibers attach to kinetochores of chromosome, and these spindle fibers that are attaching to the kinetochore sites are known as discontinuous fibers, kinetochore fibers, and the tractile fibers. And some fibers are extending from one pole to another pole, and these fibers are not attaching to the chromosomes at anywhere. These are just attaching from one pair of centriole to the another pair of centriole. So this type of the spindle fibers are known as polar fibers and the continuous fibers, as they are not interrupted by any of the chromosome. So in transition to metaphase, what we have studied two type of the fibers. One are discontinuous fiber, another are continuous fiber. These fibers are important to understand because in ncert it is just shown by diagram and explained somewhat in the metaphase stage, but what I am going to explain in the metaphase, you will get more clear about this stage in the metaphase. Metaphase is the second stage of the karyokinesis, and it is very important stage. What are the characteristic features of the metaphase? What kind of changes we are going to see here in the metaphase? Here in metaphase, complete disintegration of nuclear membrane will occur. Nuclear envelope will completely get disintegrated. It is your ncert line. Complete disintegration of nuclear envelope marks the start of second phase of mitosis. Due to this complete disintegration, chromosome spreads through the cytoplasm of the cell, that we have already studied in the prophase part as well. So one word is very important here, that is complete disintegration. If in question it is marked that complete disintegration of nuclear envelope occurs during, your answer should be metaphase. During metaphase, chromosomes are maximally condensed, and during this stage chromosomes are thickest and shortest. During metaphase chromosomes are thickest and shortest मतलब छोटे और मोटे. That is why they are very clearly visible under microscope, and due to this reason it is the best stage to study the morphology of chromosome. It is very important MCQ for your NEET. Multiple time questions has been asked from this line, which is the best stage for studying the morphology of chromosome. Then answer will be metaphase. How chromosomes appear? You have seen here chromosomes are appearing with the two sister chromatids. So chromosomes appear with two sister chromatids, and these two sister chromatids join at one common point, that is the centromere, like this. This point is the centromere where two sister chromatids are joined, and lateral to this are the kinetochores, which are the disc-like structures. So here I have written this: chromosomes appear with two sister chromatids joined at centromere. At lateral sites, kinetochore, which are the disc-like structures present, and these are the sites of attachment of spindle fibers. Again, it is very important question. What serves as the sites of attachment for spindle fibers? Then answer would be kinetochore. And why here spindle fibers are attaching to the kinetochore sites, and why this attachment of spindle fibers is important? So that chromosomes can move. Spindle fibers will help in the movement of chromosomes, right? And next important line, during metaphase chromosomes move and they get aligned at the equator. This movement of chromosome is known as congregation, right? We can say here in transition stage you can see chromosomes are scattered throughout the cytoplasm, but during metaphase what's going to happen? This is NCERT figure, I will explain it later. Here during metaphase what's going to happen? This is centrosome having one pair of centrioles. Initially chromosomes were scattered throughout the cytoplasm. Initially chromosomes were scattered throughout the cytoplasm like this. We have studied there are kinetochore fibers, which is attached to each kinetochore site with respect to each chromatid. These are the discontinuous fibers, and there is a continuous fiber that is connecting one pole to another pole. Initially they were scattered throughout the cytoplasm, then in metaphase these all chromosomes move and get aligned in one straight line, that is at the equator. You know this is the equator, this is equator, and this perpendicular or vertical line is known as the pole. So here all the chromosomes get aligned horizontally. All the chromosomes gets aligned horizontally. Just wait. This is the centromere. If we draw an imaginary line through this centromere, all the centromeres lie in one straight line, that is at the equator, and this imaginary line is known as equatorial plate, or we can also call it metaphasic plate. how these chromosomes are able to align at the equatorial plate? This was possible only due to the spindle fibers. This discontinuous spindle fibers that are making it possible so that all the chromosomes can get aligned and maximum condensation occurs, so chromosomes can be clearly visible. That's why it is the best stage to study the morphology. So you can see here centromere. Here is centromere of all the chromosome at equator, and where does arms of chromosomes are aligned at poles, right? That is the very important MCQ. You can see here in the NCERT diagram, this is the one pair of centriole, this is another pair of centriole. So here continuous fibers are not shown. That is not essential to show here in this diagram. That was just to make you understand. Here these are the discontinuous fiber. Per chromosome you can see 2 fibers. Per chromosome 1 fiber. This is the second fiber. This is one chromosome having 2 sister chromatids. Per chromosome 2 spindle fibers are attached from the opposite poles, and here all the chromosome lie at equator, and this imaginary line is known as equatorial plate or the metaphasic plate that I have made. This is the transition to metaphase, that is just after prophase how chromosomes were seen, and this is the metaphase stage. So we have studied in metaphase all the chromosomes are aligned at equator. Got it? An imaginary line is passed through the centromere which lies at equator, and arms of chromosome lie at poles. This imaginary line is known as metaphasic plate or the equatorial plate. And this plane of alignment of chromosome at metaphase is referred as metaphasic plate. That is your NCERT line. In NCERT, one another line is written: all the chromosomes that lie at equator with one chromatid of each chromosome connected by its kinetochore to spindle fibers from one pole, and its sister chromatid connected by its kinetochore to spindle fiber from opposite pole. In simple, one chromosome has 2 sister chromatids and each chromatid is attached to the centriole with the help of kinetochore fibers. So we can say just one important MCQ: per chromosome 2 spindle fibers are attached, one from each pole. This is again an important MCQ question. Right, that's done with the metaphase. So we are going to do some important questions here. In metaphase of mitosis, spindle fibers attach to, where does the spindle fiber attach? Centromere? No. Kinetochore? Yes. Chromatid? No. Telomere? No. So answer would be kinetochore. These type of questions are very important. During mitosis, the disappearance of nucleolus and nuclear membrane occurs in. We have studied disappearance of nucleolus occurs in prophase, metaphase, anaphase, telophase. The right answer would be prophase. Stage of cell cycle at which condensation of chromosome begins. It's not asking where fully condensed, it's asking where does condensation begins. Options as in question 2. So answer here would be again same, that is the prophase. So that's done with the metaphase stage. Thank you.